Okay, well, welcome, and sorry the fan's on again because it's kind of hot right now, so just gotta keep the air flowing. So, we're going to continue this review of the testis in immune privilege, and but before we do that, I want to go over a few things. So one of the papers that I want to go over later is the um, there is a preprint paper that was released by a very well cited and a very prolific researcher in virology down in Australia, and he had gone through what would it be called? Not bioinformatics, but basically computational studies of the SARS-CoV-2 spike protein and compared its binding energies or binding affinities with a bunch of different uh, ACE2 receptors of a bunch of different species. Okay, so remember from one of my first videos about binding and the structure of the SARS-CoV-2 spike protein was that the strength of the binding usually indicates the strength or the, the pathogenicity and the virality and the transmissibility of the virus. So a spike protein with a lot more affinity for the ACE2 receptor would be more associated with a much more contagious disease. And oftentimes, a more contagious disease goes hand in hand with a more virulent and pathogenic disease, although not always. So, what what they were, what this group in Australia had reasoned through computational methods. So remember, this is not through uh, like a test tube test. This is exclusively on computers using biophysics. They realized that the human ACE2 receptor was the far greatest, uh, or had the far greatest release in Gibbs free energy, which in non-technical terms means a lot of energy is released when the spike protein and the human ACE2 receptor come together. And that amount of energy that was released is far greater than that. I shouldn't say far greater, but it is the greatest out of all tested ACE2 receptors from different species. So this does not make a lot of sense because you would normally see find that a different species actually has comparable or better affinity to the ACE2 receptor, especially if it has crossed the species recently. So if it crossed the species recently, it probably would indicate that the original species is still better at transmitting the virus, but since in this particular case, since the human ACE2 receptor stands out in how good it binds with the SARS-CoV-2 spike, this would indicate that this virus had been circulating in humans for a very long period of time, or it has gone through a lot of successive duplication and mutations throughout using human cells. Now, this does raise some questions as far as how the heck can that happen? Really, that is. I'm gonna sneeze. Excuse me. How can that happen? Well, it could happen that it crossed into um, a very similar mammal to humans, like some primate, and then existed in primates for a long period of time, and then crossed into humans. But it's even better in humans than in primate So we really don't know. I will leave that to the listener to conclude what possibilities there are. There is the other possibility that we do know that this happens for a fact, where if you culture viruses in different cell types, then those cells and the viruses end up multiplying while being cultured, and they very readily undergo evolution while they are being cultured to bind to the receptors of that cultured cell more and more. So remember, I've gone over this in previous videos, but a lot of times we don't do experiments on the full living creature. We do experiments on cell lines or cell cultures, which are just uh, cells that we've taken from the organisms and we've cultured them, and it's easier to do experiments on some cells than it is to do the experiments on a full-fledged creature. And we often um, we can use those cells as a method of culturing viruses as well. So this would be a possibility. And keep in mind, there's no immune system in these cultured cells. So viruses are very able to multiply much more rapidly. They can multiply without nearly as much selective pressure, and so they are able to explore a lot more different states, genetic states, and amino acid sequence states during this process. So this would theoretically allow viruses to mutate to become better and better at binding human receptor if it was human cells that these were cultured on. So yeah, so this is of concern because the ACE2 receptor, I forgot to mention earlier, this is how it ties into this particular review of the immune privilege of the testes, is that the testes have extremely high production and expression of the ACE2 receptor. This is something that I had been screaming about from the very beginning, is that your testicles are some of the highest expressing ACE2 uh, ACE2 positive cells in the entire body. And on top of that, the germ cells, which these germ cells are undergoing necrosis, but the germ cells over here, you can see these actually are expressing ACE2. I think it's like 50-fold higher than your lung tissues do. So don't quote me on that, but they express ACE2 in such incredibly high concentrations that they are prime targets for this virus. So if you ever go through a period of viremia, which is when you have virus in the blood, those viruses will be able to transfer down into the testicles. And what is very likely is, especially during a little bit of inflammation, these tight junctions are compromised in some way. And we also know that the immune system is suppressed in the testicles as well. So if the viruses are able to get into the testicles, they have the benefit of having a suppressed immune system in that location. So what this tells me is we normally have this physical barrier that helps protect the developing germ cells, but we know for a fact that this has been compromised in SARS-CoV-2 patients. We know that the orchitis and the inflammation of the balls from autopsies, plus confirmation that a child in Brazil had shown orchitis as well. And keep in mind, oftentimes they're not even looking for this. And oftentimes you don't even know if your balls are inflamed. So, um, continuing, we know that these germ cells have been attacked, or at least the tissues around the germ cells have been attacked. And if the tissues around the germ cells have been attacked, we can be pretty darn sure that the germ cells themselves have been attacked as well. Okay? And if the germ cells have been attacked as well, we can go down to this figure. Remember, this is the figure when you have the immature dendritic cells that have been forced to mature into the mature dendritic cells, and they matured because germ cell loss and necrosis is releasing things like the heat shock proteins and the PAMP, and these tell the immature dendritic cell to go from an anti-inflammatory immunosuppressive state to a pro-inflammatory immunostimulatory state. And then these autoantigens, or the components of the germ cells, or possibly the Sertoli cells or other cells nearby, they will end up releasing autoantigens as they break down. And these autoantigens could be taken up by the immature dendritic cell, and then as it matures into the mature dendritic cell, then these mature dendritic cells will be presenting the autoantigens that are from these germ cells, and then will create autoreactive T cells, or I should say, they will stimulate naive T cells to start attacking those autoantigens. And lo and behold, where are those autoantigens at? Well, they are in the germ cells. So these autoreactive T cells are then going to start attacking these germ cells. Okay? That is a situation that we need to be on high alert for. So, because why do we worry? We worry because we might end up with severely hampered hormone levels if you have systemic or chronic testicle inflammation and testicle autoimmune responses. And on top of that, there could be infertility that needs to be considered, because there are a lot of people in this world that want to start a family. And it is expensive as hell to have a lot of people go through in vitro fertilization, if they can do that, right? So this is why I am concerned, is that we know for a fact that ACE2 expression is disgustingly high in the testicles. So we need to be on high alert about how the virus is possibly able to penetrate past those barriers. And we see that. We see that happen with the orchitis of, I should take a step back. We're not for certain that this is happening, but we're seeing inflammation of the testicles. And multiple things could cause that. It could be the result of blood clots in the area. It could be the result of other hormone imbalances, what have you. But we do know that inflammation means compromising of the blood testis barrier, and that compromise would lead to the possible infection of the SARS-CoV-2 virus. And once the virus is there, it is there to stay for a period of time. Okay. So I raised the alarm on this 5 months ago, I think. So hopefully the mainstream media will start considering it and start bringing it up. So anyways, with that out of the way, I will continue this review, and hopefully we will finish within, I think, 30 minutes, because there's not that much time, not that much left. So the mast cells, and the mast cells are a different type of immune cell as well. So, continuing, mast cells are chiefly known as the primary responders in allergic and immune reactions to parasites. However, recent studies have shown the varied and complex contribution these cells make to adaptive and innate immunity, suggesting that they are also prominently involved in the regulation of immune responses, especially the development of autoimmune diseases. So, the variety of potent pro-inflammatory mediators expressed by mast cells, which would be histamine. So, when you take an antihistamine, that is something that suppresses the histamine response in one way, which helps you with allergies or different types of uh, insect bites. If you're having a large reaction to them, you might take an antihistamine. They can also make you tired, apparently. Interleukin-4, interferon gamma, necrosis factor alpha, or tumor necrosis factor alpha, and MIP1 alpha. Just know that these are just pro-inflammatory mediators or chemicals that are released. And widespread tissue distribution of these cells make them prime candidates as modulators of autoimmune responses. So these are a type of immune cells that are found ubiquitously or throughout the entire body. Anyways, data from in vitro experiments indicate that the direct interaction with autoreactive T cells may be sufficient for mast cell activation. The induction of degranulation and subsequent cytokine production. So here, going to this, for the testicles at least, if you have autoreactive T cells, they might interact with the mast cells, and the activation of the mast cells will cause the induction of degranulation and subsequent cytokine production. So let's look up what degranulation is, because I think I have an idea, but I really need to brush up just to make sure that I'm understanding everything. Excuse me. Speaking of allergies. I'm allergic to very bad internet. Think to that. Okay. Okay, the degranulation process in a mast cell. 1, you have an antigen. Okay. 2, you have your antibodies, so the IgEs in this case. So those are there. 3, you have these are Fc receptors. So remember from I think it was 2 videos ago, we talked to, no, this was longer. This was probably 3 or 4 videos ago, where we talked about the Fc receptors, which are the receptors on immune cells that interact with the antibodies. So if there's antibodies coding a virus, the Fc receptor is, there's actually a variety of Fc receptors, but it is the receptor that is on the immune cells that interacts with the tail of the antibodies. And this had implications in the antibody-dependent enhancement that we so often see in coronaviruses, where the first time you catch a disease, oftentimes it can be bad, right? And you would hope that the antibodies that you produce from it make it so that the second time you contract the disease, you do not show pathology. But this isn't always the case. And in fact, sometimes antibodies can increase the pathology by first coding the antigen or coding the virus, but doing it in an imperfect manner. And if they imperfectly coat the virus, then the virus ends up interacting with these Fc receptors on an immune cell, but it's not enough to deactivate the virus. So what happens is the virus ends up interacting with the Fc receptor and gets into the immune cell. And then that was a way to make your immune cells vulnerable to the virus. Whereas normally it would require, for instance, in the case of SARS-CoV-2, it would require an ACE2 receptor. But for antibody-dependent enhancement, they don't use the ACE2 receptor. They use an antibody and the Fc receptor to get into the immune cells instead. So this is, like I said earlier, a virus employing multiple strategies in order to invade different cells in the body. Not all viruses utilize this strategy, but coronaviruses, as a general rule of thumb, have been. So, and this had serious implications with vaccine design because if you design the wrong vaccines that create the wrong antibodies in the body, those wrong antibodies could theoretically increase the severity of contracting the disease. Or when you do contract the disease, the severity, the risk goes up because the virus is able to infect more cells than it normally would have. Okay, going back to this. So the first, for degranulation, the first thing is you have your mast cell here, and you have your antigen, and the antigen has antibodies on it. And then these antibodies interact with these Fc receptors, right? So, and then 4 is, I'm assuming these are the granules. So 4 is these granules get exocytosed, I believe would be the word, but they're basically opening up, releasing their contents into the environment, and usually these are pro-inflammatory things such as histamine, proteases, chemokines, and heparin, which I believe is used as a, I think that might be used as a medication for SARS-CoV-2, now that I think about it. But I'll look into that later. Anyways, degranulation is a cellular process that releases antimicrobial, cytotoxic, or other molecules from secretory vesicles called granules. So yeah, these are vesicles or granules that are within the immune cell normally. So if we go to, yeah, so you can see it here. You have your mast cells normally, and then these mast cells are degranulating or releasing their contents into the environment given certain cellular signaling. Okay, let's continue. So that is degranulation and subsequent cytokine production. Upon activation, mast cells release numerous factors that may act as mediators and as such are capable of influencing various facets of disease induction and progression through the release of vasoactive amines, such as histamine. So I believe a vasoactive amine means that it works on the circulatory system. One second, let me just making sure I'm not teaching y'all some bullcrap. Strong. Yeah, that acts, so a substance that contains an amino group that acts on blood vessels and alters their permeability. So this is from dictionary.com if you want to look into it. I'm sure Wikipedia also has its own, but serotonin is also one of those. So that's good to know. So let's X that out. And cytokines. So through the release of vasoactive amines and cytokines, mast cells can profoundly affect vascular permeability, thereby opening the blood-brain barrier, which in turn provides for greater influx of activated T cells and increased inflammatory cell traffic. So this is what I was talking about earlier, where for our testicles, we do have a blood-testes barrier that protects the developing germ cells from the immune system that is in the interstitial space, if I remember if that is the right term that they use for it. So the interstitial compartment, right? So, but when we have the release of histamines and the release of these pro-inflammatory cytokines, what often happens is inflammation happens, and these cytokines will profoundly affect vascular permeability, or the permeability of the epithelial tissues wherever they are, which is great if you are a T cell or if you are an immune cell that is trying to go into an infected area and attack something. That is excellent because you want to be able to squeeze through the in between the cells, right? But this is awful if you have an autoimmune disorder, because then the areas that were protected by these barriers are ending up getting compromised by these histamines and these cytokines that are being released by the mast cells. And, you know, it's not just the mast cells, there's a bunch of other immune cells that are participating in it, but the mast cells seem to be playing a large role explicitly for things such as histamines or things that can be released through the degranulation process. Anyways, this scenario appears to be the case in human multiple sclerosis in its rodent model. And this is the experimentally EAE. That was the experimentally autoimmune encephalomyelitis, I believe. Let me EAE. Yeah, so the experimental autoimmune encephalomyelitis. So that is when you experimentally induce the autoimmune reaction that causes inflammation of the nerve cells, right? Or probably demyelinating events as well. Let me, where do we go? Oh yeah, where numerous studies have found a correlation between the number and or the distribution of mast cells and the development of the disease, increasing degranulation and elevated tryptase levels in cerebrospinal fluid of multiple sclerosis patients indicate the presence of activating mast cells, which are suspected in altering the blood-brain barrier and facilitating the entry of T cells into the central nervous system. So a few videos ago, I went over beta endorphin and how beta endorphin was used to protect mice from protect mice from demyelinating events, which caused paralysis or partial paralysis of the rear hind legs, and could also kill the mice as well. But this disease was induced by the injection of a coronavirus, specifically a murine or a mouse coronavirus. And by injecting that coronavirus into the mice, it ends up causing, brain swelling and a bunch of damage to the nerves. And this damage to the nerves causes things such as paralysis and or death, right? But if they gave beta endorphin, which is a regulator on the immune system, to these mice, then the mice were then able to, they basically cut the fatality rate in half. And out of the survivors had a less pathology as well. So this can tell you that drugs that inhibit the beta endorphin, or that drugs that inhibit the pro-inflammatory response can often stop or alleviate some of the longer-term consequences of contracting these viruses. Okay, and this can also play a role. This can also play a role in some other autoimmune diseases, such as I went over another paper where we spoke about. Oh, did I? I don't know if I actually made a video on that. I'll have to look back and see. But I might have just talked about it. But there was a woman who had what is it called? There was a case study of a woman who had some sort of autoimmune intolerance to, I believe, gluten or some wheat product, and that caused both skin lesions and intestinal discomfort, extreme intestinal discomfort. And so they cut out the wheat product from her diet, and that significantly helped her digestive and intestinal discomfort. But for years, she still had the lesions on her skin, which was also a part of this autoimmune disorder. So what ended up curing her of that was giving her, paradoxically, they gave her an opioid blocker, or an opioid antagonist. But the benefit of this was it was a short-lived opioid antagonist. And what I should also mention is that opioids work with the same receptors as beta endorphins. So beta endorphins and opioids attack similar receptors, or the same receptors. So they give her this opioid blocker, which would normally, one would think it would make things worse. But this particular opioid blocker was very short-lived. It only lasted for a few hours. So when they give it, the body ends up creating a pro-endorphin, anti-inflammatory pro-endorphin response, and that those extra circulating endorphins ended up curing her her autoimmune lesions on her skin after a few months when it took years. It was years that she experienced these lesions. So what what does this tell me? This tells me when we are viruses can very effectively take over or dysregulate the inflammatory response in order to benefit themselves. It can benefit themselves by making you leakier, which can expose more of your cells to the virus. It makes you more likely to sneeze, sniffle, have more drainage. So think of when you have an allergic reaction with pollen and your nose is running and you end up taking antihistamines in order to help with your allergies. Well, these histamines that the mast cells are releasing during degranulation are making your entire body pro-inflammatory, pro-leaky, pro-spraying, spewing, pro-symptomology. So that is one way viruses can take over this process, right? And by doing that, they end up becoming more transmissible because if you're sneezing, if you're coughing, if you're leaky, you're more likely to get the next victim infected. Anyways, let's continue. Um, indicate the presence of activating mast cells which are suspected in altering the blood-brain barrier and facilitating the entry of T cells into the central nervous system. Okay. Mast cells are derived from CD32 positive hematopoietic progenitor cells and initiate their differentiation in the bone marrow under the influence of the c-KIT ligand, which is a stem cell, for example, a stem cell factor, and the interleukin 3s. That doesn't matter too much. Similar to dendritic cells and macrophages, they undergo the final stages of their differentiation and/or the maturation locally. After the migration of their precursors into the vascularized tissues or serosal cavities, in which they ultimately reside. So I'm going to look up what serosal is that like a cavity that contains plasma? That's So serous membranes are membranes lining closed internal body cavities. Um, so they secrete a slight amount of lubricating fluid. So let's see if Wikipedia has something. It's a smooth tissue membrane consisting of two layers of mesothelium which secrete serous fluid, the inner layer that covers the organs in the body cavities. Okay. So it looks like the serosa is just in between different types of epithelial layers. I'm not going to go too into that. Just know that this is where a lot of these mast cells, or the immature form and the undifferentiated form of the mast cell, tends to localize at. Mast cells are often associated with blood vessels and are found within mucosal surfaces of the gastrointestinal and respiratory tracts in the skin, and in close proximity to peripheral nerves. The distribution of mast cells in the testes of rats, mice, dogs, cats, bulls, boars, and deer is associated with the blood vessels or close to the tunica albuginea. It's all Greek to me. One second, let me look this up. That might be Latin, actually. something in the penis is a fibrous envelope that extends the length of the corporocavernosa penis, containing the erectile tissue, and the corpus sponginosum, containing the male urethra. It is a bilayered structure that includes an outer layer of that includes an outer longitudinal layer and an inner circular layer. It consists of approximately 5% elastin, with the remainder mostly consisting of collagen. With the tunica, okay, so it's this white part right here. So this is the tunica albuginea. Okay, so this is where the mast cells in the penis tend to reside. In contrast, that's where they are in the rats, mice, dogs, cats, bulls, boars, and deer. Okay, so that's different from humans. In contrast, human testis mast cells are localized throughout the interstitial tissue and capsula, with numbers increasing slightly during infancy, decreasing during childhood, and decreasing again at puberty. The interstitial tissue and capsula. What was the capsula? We're learning a lot about the male anatomy right now. Capsula penis. Well, yeah, that's not really helpful. So just know it's a part of the penis, with numbers increasing. Yeah, in mammalian testes, mast cell mediators are involved in the regulation of steroidogenesis by Leydig cells. Okay, so the mast cells mediate the regulation of creation of steroids in your body. That is fascinating. The mast cell's main secretory product, the serine protease tryptase, acts as a potent mitogen for fibroblasts and enhances the synthesis of collagen. This serine protease that alarms me, because serine proteases are implicated in allowing the viruses to enter the cells. So if there happens to be more of these serine proteases released at any one time, that might increase the amount of viruses that are infecting cells, or the number of viruses that are successful in infecting cells. I'll have to look into that later. Which in turn can result in fibrosis, sclerosis, thickening, and hyalinization, which I believe would be an increase in hyaluronic acid polymers in the body, of the wall of the seminiferous epithelium, all common histological features found in male infertility. So all of these things can be associated with, so if the mast cells start releasing more serine protease, which, remember, proteases are enzymes that break down proteins, this can theoretically enhance the synthesis of collagen, which could cause fibrosis, sclerosis, thickening, and hyalinization of the wall of the seminiferous epithelium. And this might be something that causes infertility as well. So hyalinization is the process whereby tissue degenerates into translucent glass-like substance. Interesting. So abnormal spermatogenesis and infertility have all been associated with the increased numbers of mast cells. Well, that's problematic. I'm going to look up real quick. Mast cell ACE2. I remember? Come on, nature. I'll get back. Okay. There we go. Okay. So doesn't look like it. I'll worry about that later. Anyways, continuing. Our understanding of the role of mast cells in inflammatory disease have taken a new turn since the discovery of proteinase activated receptor 2, or PAR2, more than a decade ago. PAR2, a member of the novel subfamily of G protein coupled receptors, and I have gone over this a little bit in a previous video, but just know that these are a type of receptor that can transfer cellular signaling from outside the membrane to the inside of the membrane. And a lot of these are activated by extracellular proteolytic cleavage by serine proteases such as trypsin, mast cell tryptase, and bacteria-derived enzymes, which unmask a new N-terminal sequence that in turn acts as a tethered receptor-activated ligand. So for basically, proteases can come in, they can cleave a portion of this receptor, and the cleaving of this receptor exposes a new set of amino acids, and these can act as tethered receptor-activated ligands. Recently, PAR2 protein was localized in the rat testis up to the peritubular cells, monocytes and macrophages, and acrosome of the developing spermatids. Really? The acrosome of the developing spermatids. Okay. In rats with experimentally autoimmune orchitis, PAR2 expression was found to be highly upregulated and linked to granuloma formation and tissue remodeling. Interestingly, mast cell numbers were tenfold higher and widely distributed throughout the interstitial space, particularly around granulomas. Okay, well, this also concerns me now, too, knowing that we can have an increase in protease activity when we go through an autoimmune response. Is that mast cell numbers were tenfold higher and PAR2 expression was highly upregulated? Okay, well, no, it doesn't say anything about the protease release, just that the PAR2 was highly released. Okay, we'll keep on going. We're almost done. Numerous of these mast cells were found to be degranulated, indicating that they had and were releasing tryptase. Okay, so yeah, that is indicating that there is a release of tryptases and release of these proteases into the environment. So what does this tell me? This tells me that if you get into one of these pro-inflammatory states of the testes, which we see in SARS-CoV-2, then we have an extra release of proteases. And those proteases might be making it easier for the virus to infiltrate the cells, because these proteases are, I have another video on how proteases are necessary often for viruses to get into the cells. What I'm seeing here is that we can have an upregulation and a higher secretion of these proteases during the orchitis. And we see orchitis in people who have died, like in the autopsies, we see orchitis. So this tells me if you're unlucky enough to get orchitis, then you're probably going to have a lot more viruses entering your cells, which could increase your viral load even further. Interesting. Okay, we'll keep on going. The increase in numbers and the redistribution of mast cells away from the local location exclusively under the organ capsule are important findings, as they mean that in experimental autoimmune orchitis, mast cell tryptase and its receptor PAR2 are now in close proximity, thereby facilitating an interaction between the two effectors. And you know, when I read this, I think that's not just the two effectors. That is going to also be abused by a virus. A virus can see this interaction, or what I should say is viruses have probably experienced this interaction before. They've experienced throughout their billions of years or hundreds of millions of years interacting with animals. They see this happen, and therefore they take advantage of it. And we can see, at least in SARS-CoV-2, they've taken advantage of it. So yeah, we can continue. What could be the consequence in the testis? In vivo, experimental injection of mast cell tryptase caused elevated expression of key inflammatory mediators, MCP1 and TGF beta 2 and cyclooxygenase 2, or COX2. I believe this is what aspirin and COX inhibitors work against. So anti-inflammatory drugs, I believe, often work on this. Largely in PAR2 dependent mechanisms. In vitro studies indicate that peritubular cells are responsible for the observed increase in MCP1, TGF beta 2, and COX2 following PAR2 challenge. It was hypothesized that the release of MCP1 could at least partially account for the observed mass recruitment of inflammatory monocytes and or macrophages to the chronically inflamed testes, which have not yet adopted the testicular phenotype. Similarly, also the above-mentioned testicular fibrotic disorders were related to PAR2 activation by mast cells in a COX2-dependent mechanism. So what this is saying to me is that this is a positive feedback loop that could result in permanent damage if it doesn't get controlled in the early stages. So you can do an initial injection of these something that is pro-inflammatory, and if it can dysregulate the mast cells in this way, they start releasing more of these pro-inflammatory cytokines, and then that could result in things such as fibrosis, or they had a list of fibrosis, sclerosis, thickening, and hyalinization of the walls of the seminiferous epithelium. Well, crap, if you have that happen here, you can induce those types of things by just giving them the protease. Let me read this or the tryptase. So if you inject the mast cell tryptase, you can create all these downstream inflammatory processes, and then also these downstream pathologies just from the tryptase of the mast cell. So you do not want your own mast cells to be releasing high levels of tryptase, because then you might see this happening. You know what this tells me is that, with the other human coronaviruses, I don't think we see the level of orchitis as we do with SARS-CoV-2, MERS, and SARS. So what I'm guessing is going to happen is that if we let this disease run through society, we're definitely going to see, I'm 95% sure about this, we are going to see accelerated levels of infertility in people, and as a result, lower testosterone, dysregulation of hormones, sex hormones specifically, but other hormones as well. So yeah, I'm not too happy about this. Similarly, also, the above-mentioned testicular fibrotic disorders were related to PAR2 activation by mast cells in a COX-2-dependent mechanism. Okay, so maybe taking aspirin might help. I don't know. That'd be to help prevent that. Maybe that's something a doctor would probably want. So that's something that a doctor might want to look into is if they start seeing inflammation of the testicles of some of their patients, um, they might look into COX-2 inhibitors after they've recovered from the coronavirus. Maybe the administration of an anti-inflammatory after the fact might be beneficial. We'll see. Anyways, we are only beginning to understand the role of mast cells in testicular immune privilege and inflammatory disease. It appears, at least in the normal rat testes, that the very restricted distribution of mast cells and the relatively low number is a mechanism meant to physically separate them from the PAR2 positive cells and the T lymphocytes, as drugs believed to stabilize mast cells have been shown to ameliorate the severity of EAE, which is that encephalomyelitis. Excuse me. And as mast cell blockers have been shown to be beneficial in the treatment of idiopathic oligozoospermia and oligoasthenozoospermia. Prevention of mast cell activation may be an important factor in the maintenance of an immunosuppressive phenotype, not only in the testes, but also in other immune privileged sites. So, okay, I feel like I'm talking out of both ends right now, but this is why I'm worried. Is it's almost like a catch-22, right? So your immune system is suppressed in the testes, right? Normally. But there are instances where the immune system can't be suppressed in the testes. But if the virus ends up dysregulating this, you could end up with an inflammatory response, which can cause damage. But even with that, the immune system is still compromised. It is still trying not to attack. So that could allow a virus to last a lot longer. That could cause a virus to last a lot longer in the testicle. So it's almost like you're screwed. You're damned if you do, damned if you don't, right? And if we have this immunosuppression, if normally a healthy individual might just say, screw it, we're not going to have the immune system function in this area, well, then a virus can take hold very easily. Or then they could decide, okay, we are going to start inflaming things to try to get rid of the virus, but then that causes damage to the testicles. So it might be that it cycles back and forth what to do depending on the viral load. And the fact that the germ cells are so incredibly sensitive to the, or I should say, the testicular tissue, especially the germ cells, have such high expression of ACE2, that would result in high susceptibility to the virus. So it'd be very, very, very difficult once a virus got in there to be removed. Yeah. Yeah, that's... Which, I mean, it could possibly be removed after enough time, right? It could leave. It could eventually be taken care of. But what if you're still contagious through your seminal fluids during that entire time, right? What if it takes a year and let's say you are prolific in your in the dating scene, right? Then you theoretically could be exposing a lot of people to contaminated seminal fluid. There's semen, and you might have recovered completely in the other parts of your body, but if your seminal fluid is a place of concern, you're not necessarily going to know. And so that, and the fact that oftentimes when it comes to when it comes to normal sex, at least very often um, we do safe sex practices in this country. At least we try, right? So if it's not like a long-term partner, very frequently we try to have at least with normal. I should say normal, but when it comes to vaginal sex, we often try to um, use condoms, right? We try to protect each other from the exchange of fluid when it comes to vaginal sex, partly because of the risk of pregnancies. We at least attempt that. But yeah, now I'm when it comes to things such as oral sex, we we almost never we almost never utilize some sort of dam to prevent that from happening, prevent the exchange of fluids in that case. So yeah, I definitely could see that if the semen does end up being contagious long after, um, and if we do start returning to a more promiscuous society after all these lockdowns are ending, um, we could end up seeing a resurgence of people who were sick actually months ago, right? Like people might have been sick a long time ago, but if their seminal fluid is still contagious later, then we might see a spike later and associated with more promiscuous behavior, which like after lockdown, there's going to be a lot of people who are going to be very excited to be back in the hookup scene. And so when this ends up happening, you could have a bunch of people, especially if someone had this disease, and assuming that they still have a very high sex drive, yeah, they're very likely to go out and participate in the opening of the country. So yeah, that's one of my concerns. I should say is that uh, damned if you do, damned if you don't, almost. Anyways, a conclusion. There is now widespread agreement that the immune system, spermatogenesis and steroidogenesis, the intrinsic testicular function, are intricately linked by a network of complex interactions. I should mention this is this is a very stereotypical political statement, right? It's like I should say like politically neutral statement is my field. The things that I work on are very complex, and they're like everything is a network and intricately connected, right? So that's almost one way of saying, yeah, this is pretty complicated, and it's hard to know everything because it's biology, it's a noisy field, right? So yeah, just whenever I read something like that, I'm like, you're literally saying nothing. You've used a whole sentence to say pretty much what we all already knew, right? But anyways, the importance of the delicate balance needed between the suppression of the immune response to protect the germ cells from autoattack on the one hand, and the ability to activate an immune response to prevent damage from infection, trauma, and cancer on the other is reflected by the fact that in the human male, about 12 to 13%, in some studies even more, of all diagnosed infertility is related to an immunological reaction, immunological reason. So about, 1 out of 8 infertility cases are associated with an autoimmune or an immunological response. While its contribution to idiopathic infertility, 31% of all cases, remains unknown. Wait, that in the human male, idiopathic, I think, is when they don't know. Let me double check that. I think it's when you don't know. Yeah, so it's an unknown with unknown cause. So in 1 out of 3 people with infertility, they don't know the cause of infertility. So there is a possibility that a large number of these unknown infertility cases is associated with the immune response in some way. That's what they're saying here. Anyways, the 12 to 13%, that is just when that is what's diagnosed, when we do know what is causing it, and that cause is the immune system. Okay. The mechanisms responsible for the testes immune privilege are still far from being understood, but it's apparent that the identified factors involved are multiple and probably redundant. And this is something that I like to see, redundancy. Redundancy happens in biology all the time, where you will employ multiple strategies to get the job done. Because if one of those strategies fails, you still have redundancy in a bunch of other systems to make sure that you still get the job done, right? So this was seen in immune privilege, where it looks like it's both a physical barrier and the immune cells and the testicle cells all secreting different chemicals that all try to protect the testes from inflammation. And that dysregulation of that can cause serious harm, right? Overall, the long regarded as a peculiar side issue of testis function, immune privilege is now established as part of the general scheme of male gamete formation and successful reproduction. So this is no longer just a fringe hypothesis. This is now front and center of how sperm survive and how it's generated. The immune system plays an absolutely intricate role in that. Overall, further research in the area will not only help to improve diagnosis and treatment of immunology-based male infertility, but also will open new avenues in contraceptive development and transplantation medicine. I don't know about. Well, I guess we could learn some contraceptives, but I personally would not want to use the immune system as a form of contraceptive. Just speaking on the other complications that we can see when the immune system becomes dysregulated. But if the scientists think that that is one of the possible things that can be gleaned from this, then they can do that research, I guess. So anyways, 1 hour and 4 minutes. So we're done with this, which is great because 3 videos on one subject it's a little much. But what I liked was this sort of shows to me the importance of immune privilege, the importance of how the immune system works, for one. So it's always good to be reminded on all the different types of immune cells that exist, because it is a pain in the ass to remember all of them. Like it really is a pain to remember all the different letters and the positive number, CD number positive, CD number negatives, like the different chemicals that they all release. So it's always good do a refresher. Um, and then, but it was also interesting to see how it can become dysregulated. How can a disease be caused when this immune privilege is lost or taken advantage of? And how the fact that this immune privilege exists in the first place, how that can cause sensitivity or long-term contagion to exist within the testicles. And maybe it's only a minority of people, right? Maybe only 10% of men. I mean, we could go with the 80-20 rule. Like only 20% of men have long-term contagious semen, right? Well, then if they're contagious for the rest of their life, or if they're contagious for a year, there there are many people who have a lot of sex partners over the course of a year, over the course of 2 years, and so we know that was it almost 4 million people have been infected already, uh, guaranteed infected. So the real number could be far greater. Half of them are male, right? So we could see, I should say about half, um, 2 million, 2 million men. How many, what percentage of those men are going to be sexually active? Well, what percentage of those men are likely going to be super spreaders, for lack of a better term, but I should say uh, prolific in their sex life? There's probably going to be hundreds of thousands of men that will be sexually active with multiple partners over the course of a year, 2 years. So, what percentage of those men will be contagious over that time? If it's 20%, that's still tens, 20s of thousands of people. So this is just something that we need to be highly cognizant of when as things do open up. So okay, um, I think that is everything that I wanted to say. So hopefully, um, yeah, hopefully you all have a good evening. Stay safe, and hopefully I will see you tomorrow.